Okay, I think we'll get started now. Chaitra.
Okay, so everyone, welcome to the Science Seminar brought to you by Youth Student Advisors and the Harvard Undergraduate Microbiology Society. Right now, we're going to be introducing ourselves and then we're going to be getting into a presentation of what our lab today is going to be.
Hi everyone, welcome. I am Vandika Doon. I am a Director of Physics for the Youth STEAM Advisors, Ambassador of Trinidad and Tobago, and a HUMS Junior Fellow.
Hi, I'm Chaitra Annampudi, and a I'm a founder of Youth Team Advisors, and I'm also a junior fellow of HUMS.
Hi, my name is Vinaya Ainampudi, and I am also co-founder of YSA and a HUMS Junior Fellow. I like exploring biology and how technology and they both intersect. Okay, so now we'll talk a little bit about the lab.
Bacterial transformation. Bacterial transformation is the process where bacteria take up foreign DNA from their environment. This can be done naturally or chemically by processes like heat shock, which we will be doing in the virtual lab today, and electroporation.
And during this process, a foreign DNA is introduced to the E. coli using a plasmid which has the RFP gene, which makes the protein glow red, ampicillin resistant gene, and the overall goal is to make the bacteria glow red and so that it can survive on the antibiotic plates if the transformation, which we'll do in the beginning of the lab, is successful.
Creating competent cells. E. coli are made competent by using calcium chloride, which is used to neutralize the negative charges on the cell membrane. This makes them ready to take up the plasmid, and the DNA and cells are mixed together in a tube.
Following this process for the heat shock, the temperature is set to 42 degrees Celsius, and after that, it's followed by a rapid cooling in the ice bath, which you'll see in the lab. And the heat shock allows the plasmid to enter the cells, which is the overall goal of this lab.
Gene expression, recovery, and selection. The recovery phase, which is after the heat shock, the bacteria are transferred to a recovery medium, which gives them time to begin expressing the gene. It is very important to be done before plating.
And for the plating itself, the cells will be spread onto an agar plate containing the ampicillin, and then the transformed bacteria, which we'll do in the beginning of the lab, will grow, and the RFP gene will make the colonies glow red. So now that we told you a little bit about the lab, we're gonna get right into it. So Chaitra will share her screen and you guys can follow along and she'll lead the way. So let me stop sharing and I'll give you guys, oh, she'll give you the link in the
Here's
chat.
the link. I will be sharing my screen shortly. So once you guys open the link, this is the page that you should be seeing. I'll wait for you guys to get to the screen. Along the way, I'll just be explaining why we're doing each step and so on. So now we're going to start the simulation.
Wait, is
So
everyone
right
on
now
the
there's
link?
three levels. We can,
Was everyone able to open the link? Let's double check.
oh yeah. Did everyone get to the link?
Oh, um,
That's okay. Okay. So now we're going to select a level. If you're unfamiliar with these topics, when we were doing the presentation, pick level 1. If you're a little bit familiar, then pick level 2. And if you're advanced, then level 3. I'll be doing level 3 today, but I'll still be explaining along the way. So right now, the context is just what was explained in the presentation. We are going to be transforming E. coli bacteria using a heat shock technique, and we're going to be using P minus and P plus um, components, and then we're going to be adding plasma to only the P plus tube. I'll give you guys like around a minute if you guys want to read this. On the second slide, it's more of the materials, what we're going to be using today, the micropipettes, the tips, the P minus and P plus tubes I mentioned, the ice bucket that they're all going to be contained in, and the water bucket where the heat shock will be taking place. You can click on each one to get more information, and I'll give you guys around a minute to read all of that. Okay, so after that, we're going to do our predictions. So we're going to be seeing what a successful transformation would be and what a negative control outcome would be. So right now, you're just going to press on which one of the tubes you think is going to be correct at the very end. It's okay if you're wrong. I will just be putting my predictions. I'm not going to say it's right or if it's wrong. So I'll give you guys a little bit of time for that. So now I think everyone is set. So right now we're going to be starting the lab, and we're going to start by setting our water bath to 42 degrees Celsius. This is the perfect temperature for a heat shock, which is what we're going to be doing today in our lab. And now, if you are on level 1, you're probably going to have way more advanced and way more like detailed descriptions. Right now, you're not if you're on level 3 because you should already know what to do. So right now we're going to take the micro pipette 200 and we're going to look at the level and set it to 50. That is how much we're going to be extracting from each of the bottles. I'll let you guys do that. And then we're going to get a tip, and that is what we're going to be using to take out the liquid. And now are the competent cells. We're going to be adding competent cells to both the P minus and P plus tubes. And you just go to the first stop on the circle above. And now we're going to put it into the P minus tube by going to the second stop. And this component is very important if you're going to be doing any lab work. You're going to have to eject the tip like for every other step because you do not want to promote cross-contamination. And now we are going to do the same thing for the P plus tube. Going to the second stop. And now we are going to eject the tip or getting rid of it. So the next thing that we're going to be doing is adding the plasma to only the P plus tube. So
Wait, so far, is everybody good with the lab so far? Like, nobody's stuck or anything?
yeah, how's everyone doing? Okay. Oh,
And feel free to raise your hand or put any questions in the chat so that we can answer them.
feel free. So I'll let everyone get to the step. I'll give you guys like around 30 seconds. If you guys are having any trouble, the other two can help you in the chat. Okay, so now I am going to move forward. So right now, we're going to set the P20 micropipette to 10 for our volume. And you guys are probably going to ask, why can't we do that on the 200? Because the lowest value that you can go for the 200 is 20, and that means we would have to take a smaller pipette. And now we're going to attach a tip. Make sure it is the right size. And if you're wondering how to open it, you just click on the tube. Maybe that is a common mistake right now, or like an issue that you guys might be seeing. And now, like mentioned before, we're going to just dispense it by going to the first stop, the same that we did before. And now only for the P plus tube, we're going to we're going to put the plasmid into it. And now we're going to switch. We're going to switch our tip. And that is how we're going to end up mixing it. So with the P plus tube, in order to mix it, you go to the first stop, and then the second stop, and then you just keep doing that. Is everyone good? I hope you guys can follow along. And now we're going to eject the tip again. And right next to it, you should see a timer. We're just going to set it to 15 minutes. This will allow the components to rest, and no, it will not take actually 15 minutes. I'll let you guys get to the step. And now we will close all of the tubes, and we'll put the P minus and the P plus into the floating tube rack, and now into the water bath. And then from here, we will give it 4 to 5 seconds for it to get the perfect time for a heat shock. And now we will open our water rack and drag our floating tube rack out. Is everyone doing good? Okay. So now we're going to take the P minus and P plus tubes to put them back into their ice bucket where they can rest again. And now we're going to set a timer for 1 minute. Now we're going to take our micropipette 200 again, and we're going to set this time to 150 for the volume. And then we're going to open the tip box, and we're going to be taking the LB broth. And we're going to go to the first stop, and we're going to open the P minus tube and put it in there. The LB broth helps the plasma grow, and we're going to get rid of the tip, attach a new one, and then we're going to mix it. We're going to the first stop, and second stop. And then we will eject the tip. Are we good so far? You doing good? So I'll let you guys get to the step. And now we will be doing the same with the P plus tube. So we're going to go to the first stop. And if you guys didn't notice, in the micropipette, you can tell when there is solution or not because it should change color. And if you look closely, when I let go, it goes away. And then we're going to have to mix it. It does not mention it, but you're going to have to mix it. There. And eject the tip. Now, we're going to set it for 15 minutes again to let the components rest, since now we just mixed them. Has everyone gotten to the step? Feel free to ask any questions. And now we are done with our experiment. These are the actual results. Um, if you guys want more accurate results, there will be another link posted where you can actually plate them and look at them. So right now, my predictions were right, and my actual results were all over the place. I have done this lab many times. It gives me almost the same result every time. You can read through your results, your predicted results. Um, and now we're going to do a reflection. So this reflection is different for every single level. And I will not be doing these because you guys should try doing them. It is a very good way to learn. So it is a review or a reflection, and we will be going over right answers. You guys do have 2 attempts for each question. There is 5 questions on each one, and we will explain it at the very end if you guys have any questions. And after that, you can go to a summary, and you guys can look at that for as long as you want.
Did
So
you
now
want
we'll
to
be taking
walk
a few
them?
questions.
Oh, did you want to walk them through the review questions, maybe? Like if you
Okay,
just
yeah.
all the levels have different review questions, but this one for level three, um, she can walk you through this one if you guys did level three.
So E. coli cells are competent, and we're going to see what that means. So it's either are these cells healthy and able to replicate, and are there small fragments of extracellular DNA, or are if these cells are able to take up large sequences of DNA. So since E. coli bacteria is what helps things grow and will help take up small fragments, it would be B for the first one. For the second one, how does the P minus tube differ from the P plus tube? So if you remember back to our experiment, we only added plasma to the P plus tube, and the P minus tube got the LE broth along with the P plus tube. So the P minus tube does not have the plasma. So P minus has no plasma, well, it and has competent cells because we added competent cells to both the P minus and the P plus, while the P plus contains both. Now, why are competent E. coli cells used for bacterial transformation instead of the wild type E. coli? So, bacteria can either take up DNA from the environment that occurs with very low efficiency, and genome DNA can be removed from competent cells to allow space in cells for plasma DNA to make them very efficient to take up DNA. And wild E. coli cannot be cultured in a lab. So for this one, I will be taking a guess. We will see. Yeah, that should be the right answer. Anyways, the environment has a very low efficiency in nature. So bacteria, so E. coli, when it's competent, it has been kind of transformed in a way where you can use it better in labs and it's more efficient. Now, why are the cells placed back on ice after a heat shock process? So a heat shock is like if you get a burn, you have to cool it down. And it was kind of like that for the fluids. And right now, it's just to let them cool down. And since they were under stress, the cell membranes were temporarily compromised. So they needed to recover. Why is it important to work in a sterile matter during this lab? So this means like, why is it important to work in like a very clean condition, kind of, and with each steps? And we had to constantly remove our tips to um, remove the chances of cross-contamination. So right now, to prevent DNA to contaminate the process, transform, to prevent the transfer of bacteria to the environment, and to help the growth, to prevent growth of non. So it should be this. Oh, yikes. It would be the first one. My bad. Okay. So now we will, so this question should be different for everyone. It's feedback. The last one, if you're concerned, trust me, I did not do that, the review before this. So I was kind of just on the spot. But so how are we feeling with this lab? So these are main things that you should be looking at right now, the summary. So the summary will help you if you're going to be doing in-person labs for your research projects, for science fair, or any other like things that you're going to go into a lab for. It is important to be clean. And while you're doing transforming bacteria, if it's not for E. coli bacteria or with P minus and P plus, it will be different for everyone because each cells have and DNA mixtures have their own type of like process to transform to different ones. And so you guys can go through this. I will end the simulation now and stop sharing.
I just want to say in the summary, that I tried to show on the last slide, there's like some of the things could be related to the recap little quiz to the end there, like for example the heat shock. So if you want to like reattempt the quiz at any time, you could probably read through the summary first and then go back.
So now we'll be taking any questions. You guys can raise your hand or you can put it in the chat.
My question is, what is the difference between RFP and GFP? Is there an advantage of a red color instead of a green color?
Yeah, for this specific lab, they chose to use the RFP gene because they both have like different properties, but like the RFP gene has like more deep tissue imaging because it has a longer wavelength, so it's easier to like image through it. And also it causes like less damage to the cells, which is what we're trying, like we're transforming the cells, we're not trying to damage them, so that's why we use the red one, not the green one. Also, this lab has a second part. It's called, I don't remember what exactly. Let me double check. But there is a second part for this. So if anyone's interested, you guys could do the second part as well, which like correlates to transforming the bacteria. I'll try and put the link in the
Yes,
chat.
it's plating transformed bacteria. So right now we just transform the bacteria and it's plating it. So I
Yes.
will send it to everyone. It is right here. This is the second part. Um, Based on this one, we will see if we're going to be doing another one and a second part while doing this lab. And hopefully you guys can join for that one too. So if you guys have any questions, like Raphael, we are feel we will feel free to.
Could I maybe ask another question? I didn't quite understand what a competent bacteria cell is. So as far as I understand it is you have to put the bacteria into a calcium chloride solution that there's a negative so that the Ca plus ions kind of make it easier for the negatively charged plasmid DNA to enter the cell, which is also negatively charged by the phosphate group. And then you do a heat shock that to make the pores larger. But what exactly is different in the bacteria itself?
Peter, would you like to answer or should I answer? You're on mute, just saying.
So competent cells, like you said, are covered in calcium chloride, and they kind of act like a wall around the DNA to kind of enclose it, and it also helps grow. So when we're adding the plasma into the P minus, no, the P plus, only the P plus, it helps it kind of mix and like helps it like a bond. Like if you're adding three things together, it helps it like stick together. And when you're adding the LB solution, it's the LB solution also helps it grow and kind of like um neutralizes it so that it can recover from the heat shock since we did it after the heat shock. It would the competent cells are kind of just like those cells that you would add in like during a science fair project. So our P minus is going to be like our control and our P plus is going to be like our experimental. So our competent cells are something that we're going to add to both. And our plasma is just going to be added to one so that you can see the different results. And the competent cells kind of enhance how you can see the results, if that makes sense.
Can I just add in something here? At the beginning, how we were saying that there are like the competency could be cells could be naturally competent or you have to like perform a chemical experiment to make them competent. So like the competency of the cell really depends on how it is able to take up DNA from the environment. So some cells are naturally competent, meaning that they could just do that on their own. They don't need any help to do that. And then the chemically competent cells, which is based on like the heat shock, the electroporation, they need help. So that's why we perform the heat shock to like, as you were saying, Raphael, to open make little holes on the membrane so they could enter. So they need help to take in the DNA. So that's why they're chemically competent, and the naturally competent ones, they don't need any help. They could do it on their own.
That was a great question. Any other questions?
Whether is some application of this lab technique?
Can you repeat that question? I'm sorry.
So what are some application of this lab technique? In which fields can it be used, and what outcome do scientists want if they perform this experiment?
So what I'm getting is how can this application be used for different types of experiments? Okay. So this one is kind of specialized for just transforming bacteria using E. coli cells, competent cells, and the P minus and P plus bacteria with an LB solution. So it would be different for all of them because um, each property has different like components that like would make the lab vary. So if we had to like change an LB solution, there would be a different amount that we would have to take and put it in, and it would give a different result too.
And
So
also,
this
like, this
one
is like
is
the
pretty much specialized for just this.
And also, like, this overall process is used for a lot of experiments, like producing proteins and even DNA cloning. Like, this is one of the bigger, it's like a fundamental process that you should learn. Even though we did it specifically for E. coli, you can use it for others as well. And mainly it's used for like genetically modified organisms and other such sorts of experiments.
And is it then possible to extract the DNA that has been multiplied in the bacteria?
For this specific one, I don't think that was the purpose. It might be possible, but that wasn't the overall goal. We were just trying to transform the bacteria for to use the bacteria, but I'm not sure if you can extract the DNA.
Thinking like maybe it can be possible in the case of like insulin, like to create, human insulin or like that genetic engineering model where you extract the gene of the insulin creating gene from humans or any type of animals and put it in a plasmid as like a vector to then like transform that gene to produce the insulin. So maybe in that case.
Are there different types of plasmids that could be used for this experiment? For example, a PGLO plasmid, or are there any other plasmids that are suitable for this kind of experiment?
I mean, there are different types. For this specific one, they used that, but you could use other types for sure. This one must have been the one most suitable for the E. coli and the overall process. And I know there are some other ones like PBR322. I think that was one. And there were a few other ones too, but the one that we used for this one was not. Oh,
Yeah. Can I ask a question? Or so I was wondering, are there any other alternative uh things other than using a heat shock? And how could they be used? Or why is heat shock the main thing we want?
Peter, do you want to answer that?
So a heat shock is what we used for this transformation. It would depend on like what you're using, but there would be circumstances where you would have to chill it, like make it in cooler solutions. And sometimes, like for this one, we're doing a heat shock, so we're making it go to a higher temperature since this original one was in ice. So it's kind of like it depends on the process. But yes, there definitely are different types depending on the lab that you're trying to do.
Just to add to what Chais was saying, there's also the electroporation that we talked about like earlier, where you use like to accomplish the same thing of the heat shock to create like little holes in the membrane to make them competent so they could absorb DNA. The electroporation, you basically use an electric current to make holes in the DNA to absorb the bacteria, to absorb the plasmids.
Could this experiment be applied to, for example, mammal cells, or human cells? So I think the problem is probably that we had to kind of change or transform the reproductive cells to kind of, yeah, but is it even possible?
You mind repeating the question? Sorry. It's a little.
So I was wondering if it be possible to do the same or a similar experiment in human cells. So I think the problem is that in humans, since you have to do it with reproductive cells in order to yeah increase the amount of transformed organisms. But is it even possible gene editing using this technique in plants or in other animals that are not bacteria?
It should be possible as long as it's like a foreign DNA. This specific one, we used the human DNA, but mammal cells usually don't go under like bacterial transformation the same exact way as the human DNA does, but it could still be done. So, just a warning, there's like 1 minute left on the meeting. Oh, how am I? Yeah, so the ideal, for the question in the chat, the ideal temperature is 42 degrees Celsius, and that's what we used for this one. And how it might have been, like, how it might differ is like the results and also the overall, like, transformation of the bacteria would vary based on the temperatures.
To add on to what she said, if it's any less or any more, it wouldn't exactly be a heat shock since 42 degrees Celsius is like the perfect amount that is used. If it's below, it wouldn't be a heat shock. It would be too cool to be a heat shock. And if it was more, it would potentially burn the DNA.
Yes. Okay, so we might have to end the meeting soon because there's
Yes.
only like 20 seconds left. But we will send out the certificates, and if we don't have your full name, then we might request it through Gmail.
I
Um,
have their full names
oh,
listed.
perfect. Okay,
Yeah.
perfect. Okay,
Thank you all
other
so
than
much for joining.
thank you so much for
See you later.
joining.